Hello students. Let us today study about Avery, McCarty and MacLeod's experiment where they carried out experiment to prove that DNA was the genetic material. Before them, in 1928, the British medical officer Frederick Griffith had performed an experiment on bacterium Streptococcus pneumoniae to prove the transforming principle. Later, in 1944, the US microbiologist who were working at the Rockefeller University in New York, Oswald T. Avery, Colin M. MacLeod and Macklin McCarty, they gave the first evidence to prove yes, DNA was the genetic material. The transforming principle which Griffith stated was nothing but DNA. Let us see how did they perform this experiment. They purified proteins, carbohydrates, RNA and DNA from the smooth strain of the Streptococcus pneumoniae bacteria, the deadly, the virulent, the pathogenic encapsulated bacteria. Then they inserted these proteins, carbs, RNA into test tubes and started adding the rough strain, the harmless strain, the non-virulent, rough, non-pathogenic and non-capsulated strain of Streptococcus pneumoniae or Diplococcus pneumoniae into these test tubes. So they added this and waited for some time. Then they injected the first solution from the mixture of proteins and the rough strain harmless pathogenic non-pathogenic bacteria into mice, and to the observation the mice survived, no pneumonia developed and the mice survived. This proved that protein was not the transforming principle. From the second test tube in which carbohydrates were taken, they took this mixture, they injected into the body of the mice. Again, they saw that mice survived, no pneumonia developed. Thus, it was proved that carbohydrates are not the transforming principle. From the third test tube, where they had taken RNA with the mixture of the rough strain harmless one, they injected this mixture into the body of the mice. Here also, no pneumonia developed, mice survived. This proved that RNA was also not the transforming principle. In the fourth test tube, they had taken DNA with the rough strain. They injected this into the body of the mice, and to their surprise, pneumonia occurred to this mice, and the mice died. The mouse died. This proved that yes, DNA was the transforming principle. Now they again performed more experiment where they discovered the effect of protease and RNAs, and they saw that how these enzymes did not affect the transformation. So they took the rough strain, the non-virulent, rough, non-pathogenic and non-capsulated R type of Streptococcus pneumoniae, and they mixed it with the heat-killed, the smooth type, the pathogenic one, the encapsulated one. This mixture of the living rough non-virulent and the dead smooth virulent, they to this mixture they added protease. What is the role of the protease? The protease will digest all the proteins in this mixture. Then they injected this solution to the mice and they found that the mouse died. From this, they concluded that protein had no role in transformation because though the protein were digested, pneumonia had occurred in the mice and the mice died. Then to this, they added RNAs. Again, they injected it to the mice. Again, the mouse died. From this, again, it was proved that RNA is not playing any important role in the process of transformation. In this way, they carried out for carbohydrates also, and they found that all these three did not play any important role in transformation. When they added DNase enzyme to this mixture, this DNA is it digested all the DNA. When it digested the DNA and this solution was injected into the body of the mice, no pneumonia developed and mice survived. This proved that yes, DNA was the transforming principle because when DNA is added, DNA is broken down, ruptured, and the smooth one are not able to transform the rough into the smooth type, and thus the mice it survived. Thus, this experiment proved that only DNA was the transforming principle. So, students, I hope you have understood the experiment. Please revise it again. Thank you.